Hi everyone, and welcome back to our cells class. This will be the second class on cells, and today we will be discussing the fluid mosaic model, which is a model showing how ions and other substances are transported through the cell membrane, right? So we know the cell membrane is a semi-permeable layer, but we didn't go into detail back in Form 5. But now we are going to see exactly how it is a semi-permeable layer. Okay, so what does fluid mosaic model actually mean? It simply means that the cell membrane consists of a bilayer of phospholipids, so we call it the phospholipid bilayer. Remember, we did phospholipids already, and there are proteins floating all through the cell membrane. So all these here are proteins and different substances. So it's like a mosaic, which means it's it's a random arrangement, assortment of different things, right? So phospholipids could slide past each other because they are connected via hydrogen bonds. And remember, hydrogen bonds can unbind very easily and reform the bonds very easily. Okay, so what does the fluid mosaic model actually contain? Well, you can see it here in this image, all the labels, right? Contain a lot of different things, and each of these things have different functions. So the phospholipids form a bilayer with their heads positioned to interact with outside the cell and inside the cell. So for example, let's see that over here is outside and over here is inside. So the purple is the head of the phospholipid, right? And then we have the tails, the hydrophobic tails coming together at the center, right? So you can see the heads positioned to the outside and some of the heads positioned to the inside. Okay, so the center now, because the hydrophobic tails are positioned at the center, they actually can stop water soluble stuff from moving through the cell membrane because they are hydrophobic, right? They repel water. Okay, so there are many different proteins in the membrane, right? There are glycoproteins, which you can see here, and these are involved in cell-to-cell recognition. So this is like antennae, right? So the antenna of this cell could interact with the antenna of another cell, and these two cells can communicate with each other, right? Now there are also glycolipids, right? Glycolipids, they are also really involved in cell-to-cell recognition as well, right? So more antennae there. Then there is cholesterol. That little green thing there is cholesterol. So the cholesterol allows it interacts with the aqueous medium as well as the cytoplasm, and the cholesterol essentially contributes to the fluidity of the membrane, meaning it's not rigid like a piece of wood, rather it's like a more flexible thing. That's why animal cells can be a circle, right? Because the membrane is kind of flexible, right? So that's what cholesterol does. All right, and then there are channel proteins and integral proteins, also known as voltage-gated proteins, and we'll do more of these when we go into transport across the cell membranes, which we'll do now. So there are many different ways in which materials can be transported. There's diffusion, we know about diffusion. Then there's protein-assisted transport, also called facilitated diffusion. Now you may not have heard about facilitated diffusion, right? But before we go to facilitated diffusion, let's just remind ourselves what diffusion actually is. Alright, so diffusion is essentially the movement of molecules from an area of high concentration to an area of low concentration. Right, and there are many different factors affecting the rate of diffusion. I have this all in a note right down here, right? So you don't need to worry about missing it, okay? But the real key is protein-assisted transport via the channel proteins and the voltage-gated proteins. Right, so let's take a look at the channel proteins first. Now they have hydrophilic groups, right? Hydrophilic means water loving, right? And they line the inside of the channel. So therefore, molecules that are water soluble or ions can actually pass through these channel proteins because they are water loving. So any water soluble molecules can simply pass through the channel proteins. However, each protein is specific to one certain particle. So for example, potassium, the ion potassium, needs a potassium protein channel to pass through, right? So if there's a sodium protein channel, the potassium can't pass through there, Okay. Now, carrier proteins are a little different. They change their shape to allow certain molecules to pass through, only when certain conditions are met. Right? So, for example, there's a binding site over here, and we'll have to wait for a substance to bind to this binding site, and then that'll be like a key opening the lock to the gate, and then the gate will open out so that molecules can pass through. Okay? Active transport. You would have heard about active transport already. So let me go down to another diagram here that shows active transport right over here. Right? So active transport, unlike diffusion and facilitated diffusion, is the movement of molecules against the concentration gradient. So remember, diffusion was down a concentration gradient, right? So in order to make this happen, we need energy to push the molecules across the concentration gradient, right? So the energy is in the form of ATP, right? So for example, this could be seen in certain carrier proteins like the sodium potassium pump, which is the example that we have here, right? So as you can see, the carrier protein here, number 1, has a binding site for a sodium ion, right? So when it binds, this actually causes ATP to split, right? And when ATP splits, it releases energy, and it would allow the carrier protein to change shape and release the 3 sodium ions. Right? Now when it changes shape, it will allow potassium to bind to these other binding sites here. Right? And then again, the reverse reaction will happen, and then the carrier protein will open back out, and then the potassium will go inside the cell, and so on. It will go in a nice little cycle. That's why we call it the sodium potassium pump. Okay, so it all depends on ATP. Right? I have the note right here, active transport. Okay, and finally we have membrane assisted transport. Now this is a new one you definitely would not have heard about. Right? So let's go here. Right? We call it endocytosis and exocytosis. Right? So membrane assisted transport is where substances are moved in and out of cells using vesicles. Right? So anytime something is surrounded by a membrane, we call it a vesicle. And these occur in molecules that are too large for diffusion and active transport, right? Like proteins. Okay? So there are 2 major types, endocytosis and exocytosis. So let's do endocytosis first. Now there are 2 main types. We don't need to worry about these 2, right? We're worrying about phagocytosis and pinocytosis. So phagocytosis involves large particles, right? Now the cell membrane kind of surrounds the large particle and then pinches off here and makes a nice circle, and we call it a phagosome. Right? So therefore, the large particle has entered the cell without having to undergo any diffusion or anything, right? So the membrane actually surrounded the large particle and made it a part of the cell. Alright? And this we can see in white blood cells actually do this to get rid of germs. Right? If there's a bacteria, the white blood cell will surround the bacteria, then the bacteria will be in the white blood cell, and the white blood cell will kill it while it's in the cell. Okay? Now, pinocytosis works the same way, except with smaller particles. Okay? Exocytosis now is essentially the export of materials from a cell via excretory vesicles, right? So, something like a Golgi vesicle containing proteins like insulin or enzymes. Remember, it needs to release the enzyme, right? So, it looks like this, right? So, here we have the newly formed vesicle. It either contains good stuff or it can contain cell waste. So the vesicle travels towards the cell membrane, and the opposite happens, right? So the cell merges with the cell membrane here and opens out, and it would release whatever needs to be released outside of the cell. Okay, so before we end, let me just give you the notes, right? So here is the bilayer notes there. You can screenshot. There's your diagram, diffusion, protein assisted transport, facilitated diffusion with your channel and carrier proteins, active transport, membrane assisted transport. That's your diagram with endocytosis, exocytosis, and your diagram there. I hope you guys enjoyed this short video.